Most of the chemical digestion of carbohydrates occurs in the small intestines, where it is formed into more simple sugars like disaccharides. And this is where these large starch molecules, which are our polysaccharides, are broken down into more manageable chunks called disaccharides, which contain these two monosaccharide units. Pancreatic amylase is released by the pancreatic ducts and helps break down these large molecules called polysaccharides into disaccharides and even into shorter glucose chains, which eventually are broken down into maltose with the use of enzymes. Chemical digestion from these disaccharides into these monosaccharides is driven by specific enzymes. Maltase breaks down maltose, and we can see that here, this disaccharide of maltose, which is glucose, and then it forms our two individual glucose molecules, which can enter the bloodstream and be used by the body. We have sucrase, because we can see it's always ASE for an enzyme. This breaks down sucrose, which is our other disaccharide, containing one molecule of glucose and one molecule of lactose. One molecule of glucose and one molecule of fructose. And then this will separate into the individual monosaccharides. And eventually our lactose molecule, which is our disaccharide, will be broken down using our lactase into glucose and galactose. So you can see here OSE tells us it's the sugar or the monosaccharide, the disaccharide. And then we also have our lactase, ASE for enzyme, or sucrase or maltase. So eventually these disaccharides are broken down into the individual monosaccharides, which can be then used through absorption into the body. The summary of chemical digestion of carbohydrates starts in the mouth, where it's broken down by salivary amylase, and this is into shorter polysaccharides and even into things like maltose. We have our stomach carbohydrates, where there is reduced carbohydrate digestion, mostly a lot of protein digestion occurs in here. Once it passes back into the small intestine, this is our largest site of carbohydrate digestion, where more of these polysaccharides or even the disaccharides are broken down into the individual monosaccharides through lactase, sucrase, and maltase. And we then will have our larger small intestine, where these soluble fibers are further fermented by bacteria. Chemical digestion of proteins occurs mostly in the stomach and then eventually enters into the small intestine, where enzymes like trypsin and chymotrypsin and peptase break them into the single amino acids or even into small peptide, which are things like dipeptides and tripeptides. And these amino acids are absorbed into the bloodstream for their function in things like the creation of proteins for the body and also structural roles. The summary of proteins is it begins in our stomach. There's no protein breakdown in the mouth like we see with carbohydrates or lipids. And the stomach begins by denaturing with hydrochloric acid and using pepsin to break down the active forms. Hydrochloric acid really helps allow these gastric pepsins to break down the peptide bonds between the amino acids, allowing them to become shorter peptide chains. And once it enters into the small intestine, our pancreatic intestinal proteases, trypsin and chymotrypsin, break down the shorter peptide chains into singular amino acids and shorter peptide chains. And these intestinal peptases complete that digestion into individual amino acids, ready to be absorbed by the bloodstream for their biological functions. The last is the macronutrient, which is fat or lipids. And lipid digestion primarily occurs in the small intestine. It does begin in the mouth. When lipids are detected in the gallbladder, releases bile from the liver, and this allows an emulsification process to occur where larger fat molecules are made into smaller droplets for easier digestion. These pancreatic lipases that are released from the pancreas then help break down the triglycerides into the monoglycerides or glycerols or even free fatty acids. And these are then packaged into lipoproteins, which are absorbed into the lymph system, transported through the bloodstream near the heart for storage throughout the body. Summary of lipids begins in the mouth, where we do have lingual lipases that help break them into smaller fatty acids and chains. We have our stomach, which has minimal chemical digestion of lipids, and entering again into the small intestine, which pancreatic lipases help break down to smaller fatty acid droplets into single fatty acid chains, and glycerol monoglycerides allow them to be absorbed into the bloodstream or the lymphatic system, depending on their size. After chemical and mechanical digestion has taken place, our body must absorb these nutrients and water, with about 80% of our micronutrients and 90% of water absorbed in the small intestine. In the inner surface of our small intestine is covered in these folds that look like these hills. They're called villi and microvilli. And these are folded to maximize absorption by increasing our surface area. And with greater surface area means greater absorption of nutrients through diffusion or even facilitated diffusion or even through the process of active transport. So depending on the molecules, they may be carried from these villi into the bloodstream through diffusion, facilitated diffusion, or active transport mechanisms. Faster digesting of nutrients are absorbed in the duodenum, while slower digesting ones are absorbed in the jejunum, and the ileum. Once absorbed, these monosaccharides, which are our carbohydrates, our amino acids, which are our proteins, enter the bloodstream, while these triglycerides pass into the lymphatic system before reaching the bloodstream near the heart, ready to use, store, or further processed by the liver. The next stage of the digestive system is our large intestines. Undigested nutrients move into the large intestines, in which is thicker than the small intestine and consists of the ascending, transverse, and descending colon. Its contents include water, salts, secretions, and undigested material like dietary fiber. The large intestines is full of bacteria which help ferment soluble fiber, producing water and gas which softens stools and stimulates colon movement, helping move our fecal matter through and then able to be eliminated through the rectum. This process adds bulk to our stools through bacteria from the fermentation process, and insoluble fiber remains unchanged, further increasing the stool's bulk, preventing diarrhea, and aiding in waste elimination. Water, salts, and minerals can be reabsorbed through the intestines of the large intestine and leaving waste with minimal nutrient value left to be excreted as fecal matter or poo. The rectum and anus is the final stop, where once food reaches the rectum, digestion and absorption are complete, where semi-solid waste is then excreted from the body. The rectum and the anus muscles expand and hold waste, then relax when ready, allowing for release. Secondary organs that are involved in the digestive tract help break down food, require secretions, things like salivary glands, our gastric glands, our pancreatic glands, liver, and gallbladder. These all provide enzymes and different types of materials that aid in digestion, and they provide water and enzymes to assist in hydrolysis, which means the breaking down of molecules using water, and chemical processes which break down larger molecules into smaller ones for absorption. Examples like that are the disaccharide to monosaccharide using enzymes. So this is a bit of a summary about the gastrointestinal tract.